Thanks for nicely chairing the session. And thanks to Dr. Suresh Pusala sir also for addressing such a nice topic, important topic for this conference. Now we are having the last session of this conference, uh, which are two invited talks now. And the chairperson for this particular session is Dr. Syed Ziaur Rahman sir. And sir, this session will be for 1 hour now. Each
All right. Okay.
having 30 minutes.
Thank
So,
you, Dr. C.R. Patil. Yes. Okay. Okay. So we are already late, so we'll try to finish this session in 1 hour.
Yeah.
So thank you, Dr. C.R. Patil, once again, and good evening, everybody, dear participants, delegates, and the distinguished speakers. First of all, I would like to congratulate the organizing committee for successfully completed this conference, almost all 11 sessions on varied topics on the alternatives to animal experimentation. So, okay, so thank you for giving me this opportunity to chair this session. The last session, for this, I'm grateful to Dr. Mohammad Akbar Shah, Dr. C.R. Patil, and Dr. Sheikh Raisuddin Saab. In this session, I welcome our two distinguished speakers, Dr. Ankita Pandey from the PITA and Dr. Rakesh Kumar Singh from NIPER Bareilly. So before I request our first speaker, Dr. Ankita Pandey, to present her paper on the use of non-animal approach for pesticide safety assessment, I would like to say that Dr. Ankita Pandey is basically, as you know, that she is working as PITA's India Science Policy Advisor. As a PITA representative, she collaborates with Indian regulatory agencies and scientists to facilitate the use and acceptance of non-animal testing approaches in regulatory testing and biomedical research. So we are already aware that as a science policy advisor, so she has such duties. And good to know that she is an alumnus of the CSIR IITR, and she is also recipient of many awards, including the prestigious Tulsabai Subramani Education Trust Award, which is given by Indian Academy of Neurosciences. So I'm aware with this Indian Academy of Neurosciences award because being a member of IAN, I know this about this award. So congratulations, Dr. Ankita. And good to know that you are also an author of a very interesting book, Footprints of Toxicology in India. So Dr. Ankita Pandey, the floor is yours.
Thank you so much, Dr. Rahman. I hope I am audible and you all can view my screen.
Yes. Okay, go ahead. Thank you.
Yeah, it's visible.
Yeah, we can hear you, Ankita. Go ahead, please.
Sure. So thank you so much and good evening, everybody. At the very outset, I would like to thank the organizers for giving me an opportunity to present a talk today among all the eminent scientists throughout the world. So my talk would be focusing on the use of non-animal approaches for pesticide safety assessment. And for all of those who are not familiar with People for the Ethical Treatment of Animals or PETA India, we are an animal rights organization. And the primary area of our interest is animal protection, but it's also good science. So it's something that protects everybody. And PETA India scientists work very closely with Indian regulatory bodies for the regulatory acceptance and implementation of non-animal testing methods. So I'm sure all of you would recognize the person here. It's the former Health Minister, Dr. Harshvardhan. And similarly, we're also members of several government committees, such as the Indian Pharmacopoeia Commission, the Central Insecticide Board and Registration Committee, the Bureau of Indian Standard Cosmetic Section Committee, and Household Product Soaps and Detergent Section Committee. And we basically promote the use of technologically advanced non-animal methods to replace and reduce the use of animals for regulatory purposes, for biomedical research, and education as well. So everybody here is familiar with the principle of 3 Rs. And in yesterday's inauguration session, Honorable Vice Chancellor Professor Goel already touched upon this in more depth. So these are basically the guiding principles for more ethical use of animals in research. And the 3 Rs basically stand for replacement, reduction, and refinement. So talking about the Indian legislation, is there a provision within the Indian law to ensure that the use of this 3R principle is actively enforced? So absolutely yes, the answer is yes. We have the Prevention of Cruelty to Animals Act, which clearly states that experiments on animals must be avoided wherever it's possible to do so. And we have the CPCSCA, which is the Committee for the Purpose of Control and Supervision of Experiments on Animals. It was established under the PCA Act of 1960 with the objective to ensure that animals are not subjected to unnecessary amount of pain and suffering before, during, and after the experiments. So for this purpose, CPCSC even formulated their own breeding off and experimentation control and supervision rules that were amended in 2006. And they clearly state that replacement alternatives not involving experiments on animals should be given due and full consideration. So yes, there is a legal obligation to ensure that the 3 R's principles is enforced as much as possible. So all the speakers before me have already spoken a lot about cell culture models, 2D, 3D systems, and we had an excellent talk by Dr. P.V. Mohanan yesterday on organs in a chip. So I will not be going into the details of that. The focus of my talk would actually be on the use of these in vitro models and other non-animal approaches for the regulatory toxicity testing purposes. So we all know that regulatory agencies require a battery of toxicity tests to be conducted using animals, and this has actually been the conventional way of doing it. But for almost, for quite some decades, there has been a gradual shift in that, and that's basically owed to a lot of scientific and technological advancements that have been happening, which have fueled the development of these human relevant methods. In addition, we also have several animal welfare considerations and political considerations that have triggered a paradigm shift in the way toxicity testing is now conducted. So broadly speaking, we talk about in vitro methods. We have cell culture, 2D cell culture models, 3D cell cultures, and organs in a chip. And these models have inherent advantages over the use of animals, as we all know. Since they are human relevant, they offer a better understanding of human responses. They're high throughput, they're faster and less expensive, and of course, they are ethical. And the focus of my topic would be actually on the use of these 3D culture models for pesticide safety assessment. But before we jump into that, this is just a quick snapshot of how PETA India has been instrumental in facilitating the regulatory acceptance of these non-animal methods in India. So back in 2014, we were instrumental in getting a ban on the use of animals for cosmetic testing and household products. And since 2014, for these 2 categories of products, the use of non-animal methods is being made use of. In 2016, we had the Central Drug Standard Control Organization accept non-animal methods for assessment of ocular and skin toxicity of drugs instead of the DRACE test. In 2017, the Central Insecticide Board and Registration Committee, they updated their guidelines to include these non-animal methods, several in vitro methods for safety assessment of pesticides. And just last year, we also have these non-animal methods accepted for safety assessment of nano-agricultural products. Now, the pace with which these technological advancements are happening and how new in vitro models and other non-animal methods are being developed and validated, there is an increased opportunity for us to uptake these fit-for-purpose methods in the coming years. So talking about why it's important to prioritize the use of these non-animal approaches for toxicity testing of pesticides. As I mentioned, the Central Insecticide Board and Registration Committee that overlooks pesticide registration in India, it relies on a battery of toxicity tests which are conducted using animals. And these range from short-term studies, acute studies like eye irritation, skin sensitization, skin irritation and corrosion, to long-term and complex endpoints like developmental toxicity, chronic studies, reproduction and fertility effects. So we do not have official statistics available for India, but it's estimated that around 7,000 rats, mice, rabbits, and dogs are killed to test a single pesticide for human risk assessment. And this does not even include the thousands of animals that are killed for ecotox assessment. So of course, one of the high priority areas, why it's really important to use non-animal approaches is animal welfare. And that's exactly the reason why we all are here together, to make sure that we try and minimize the use of animals, but without, of course, compromising protection of human health and the environment. So Equally important is to be at par with the technological advancements. We have several non-animal methods that are available. These are based on either human cells or tissues, and they have they're more reliable of human responses because they better predict, they better recapitulate the human anatomic structure, the physiological systems, the morphology. And as such, since the aim is protection of human health, these models would be better at offering protection of human health than any other model. Another reason why it's important to prioritize is, of course, international harmonization. So we have several different regulatory authorities across the globe that are quite receptive towards the use of these non-animal approaches. And for India to be at par with all these scientifically advanced nations, it's very, very important to globally harmonize the way they conduct their toxicity testing. The fourth reason is, of course, we know that these non-animal methods, they're quicker, they're faster, but they also offer the advantage of being cheaper and they're more cost effective. So there's a very interesting paper that was published by the US Environmental Protection Agency in 2019. It was on reducing the need for animal testing while increasing efficiency in a pesticide regulatory setting. So what US EPA did was they did an analysis from 2012 to 2018, and they found that they basically used several non-animal approaches based on which they were able to offer waiver opportunities for certain endpoints. And many of them were subchronic and long-term studies. And because of that waiver opportunities, they were able to save as much as 300 million in the study costs and 6 million in study review costs. And icing on the cake was that approach saved as much as 2 lakh animals. So that's how we actually need to move forward, making sure that animal welfare and progressive humane science move hand in hand. Of course, without compromising protection of human health and the environment. So we've already talked about how these non-animal methods are replacing tests on animals, and this I
Something gone wrong from Dr. Ankita's side?
I think so. She has got disconnected, but still she is appearing on screen.
Yeah, sure. Yeah, she is appearing.
For a few
We'll
seconds.
wait for her.
Yes, let's wait. Yeah.
I'm sorry, I thought you were saying something. So we do have these test methods that are now available as internationally harmonized test guidelines for assessment of eye irritation and corrosion. Dr. A.B. Pant yesterday in his talk, he's already briefed everybody about these models, so I would not necessarily get into the details of this. Apart from these in vitro models, we also have ex vivo models for eye irritation and corrosion that are based on isolated corneas that are taken from bovines or from the chicken. Similarly, we do have in vitro models available for skin irritation and corrosion as well, which are being used for safety assessment of these pesticides. Now, it's generally accepted that these in vitro models cannot be one-to-one replacements for the DRACE test. And therefore, there is an absolute need to have an alternative strategy, which should, of course, be human relevant, which should be predictive, which should be reproducible. So one of the ideas could be to have a strategic combination of all these different in vitro methods, which are used in a tiered testing strategy. So just giving you an example of this, for example, if you're looking for predicting skin irritation and corrosion for pesticides, and based on the existing literature, you suspect that your test substance is going to be corrosive in nature, you use a top-down approach. So what you do is you basically start testing your test substance using test guideline 431 or 435, which is used to discriminate corrosives from non-corrosives. And if you receive a positive effect, you stop testing and you label your substance as corrosive in nature. However, if you receive a negative response, your substance may not be corrosive, but it may still have skin irritation potential. So it's important to proceed with an assay for detection of skin irritation. And therefore, you can use the test guideline 439, which is able to discriminate between irritants and non-irritants. Similarly, moving forward, if you get a positive response, you stop further testing and you label your substance as an irritant. And if you receive a negative response, also in that case, you stop testing and you label your substance as a non-irritant. Now, this was a top to down approach when you suspect that your test substance is corrosive. Alternatively, there is a bottom up approach that you would use if you suspect that the test substance is a non-irritant. And we have similar top-down and bottom-up approaches that are available for eye irritation and corrosion as well. Due to paucity of time, I will not be getting into the details of that. So this is a very interesting paper that I wanted to share with all of you. So as I said, there are several in vitro and ex vivo models that are available and are being used, but their widespread adoption is complete replacements to the Draize test. It's being hampered largely because there is a lack of concordance that is demonstrated between the results from this in vivo Draize test and these in vitro and ex vivo methods. And there are many factors that contribute to this lack of concordance. One of them is the inherent variability of the rabbit Draize test, the anatomical differences between the rabbit eye and the human eye, and the differences in the exaggerated exposure scenarios. So all of these factors result in a lack of concordance between the results that we see between the in vitro and ex vivo models and the rabbit test. So to address that, PETA, along with the US Environmental Protection Agency, we did a review of the available in vitro, ex vivo, and in vivo methods with respect to their relevance to the human ocular anatomy, the mechanisms of action which underlie the skin irritation, the eye irritation and corrosion process in humans. And what we found was that these in vitro and ex vivo methods were more reflective of human biology. They were more predictive than the rabbit Draize test. And based on that, what the gist of the paper was that since these models were more reflective than these rabbit tests, the rabbit test should not be used as a reference method to demonstrate the validity. We know that the rabbit test has been like the gold standard, the benchmark that has been set when we try and validate our new in vitro and ex vivo methods. But this is something that we need to be careful about because the rabbit test is highly unpredictable in itself. When we're comparing the rabbit test's mechanism of action, the anatomy as compared to the humans, we find that this method should not be used in a completely blind way to assess the performance and to assess the validation or to conduct validity of new methods. So there is absolutely a need to rethink the entire validation process. So to address that, PETA is closely working with the US and the EU government on another paper, which would be defining how this validation process should be, how we need to rethink the validation process, and in fact, shift the focus from directly comparing our new models to the rabbit Draize test and instead rely on methods that are actually reflective of human-based biology and approaches. So as I said before, the regulatory agency in India, CIBRC, has been relying on a checklist of in vivo studies for satisfying the regulatory requirements for these pesticide registration. However, there is a very strong need to move away from this checkbox approach towards a more weight of evidence approach. So one such approaches could be integrated approaches on testing and assessment, as we know them as IATAs. And these are science-based approaches which rely on integrated analysis of existing information coupled with new information that is generated from other techniques. So for example, if you're looking for testing your substance, instead of directly testing that on animals, the first step should be that you gather all the existing information that is available for that test substance. That could be from clinical studies, from epidemiological data, from existing in vitro and animal studies. And in addition to that, you can also use other non-testing methods that are available. For example, QSR approaches, we have read across or bridging approaches where you can actually predict the toxicity of your test substance using information for the same endpoint from another similar structurally related substance. So with all this information, all this integrated strategy together, you can conduct a weight of evidence assessment. And if you think that information is adequate enough to classify your pesticide or your test substance, you go ahead with making a regulatory decision. If no, there is, of course, a need to generate additional information, and that should be done using a sequential testing strategy. And the first step of that strategy should be to use the existing non-animal methods, whether these are in vitro or ex vivo approaches. Even after that, if you do not think the information is adequate enough to drive regulatory decisions, you conduct animal testing only as a last resort. So this is just a general workflow of a typical IATA, and we do have guidance documents on these IATAs published for serious eye damage and eye irritation and skin corrosion and irritation by OECD. Moving forward, another approach is the adverse outcome pathway. It's nothing but a sequence of cellular and molecular events that lead to a toxic effect when an organism is exposed to a test substance. And adverse outcome pathways, or AOPs, can actually be used in the framework of an IATA to drive regulatory risk decisions using mechanistic reasoning. So this is a typical workflow of an AOP for skin sensitization. We know that skin sensitization is a highly complex process as compared to skin irritation. There are different sequence of events. So this AOP uses different test guidelines that address individual events that are happening under skin sensitization. So starting from the molecular initiating event, which takes place when the toxin basically binds within the cell protein, we have two assays, the direct peptide reactivity assay and the amino acid reactivity assay, that are used to study this particular event. Moving on to a cellular response, which is taken care of by activation of keratinocytes and dendritic cells. Soon after that, these cells basically move to the lymph node, which gives an organ response. And finally, there is a response from the organism, which is skin inflammation. So this is a typical AOP for predicting skin sensitization. Right. So this was just like a quick high overview of what the existing non-animal approaches are that are being used for pesticide safety assessment. Moving forward, I would just quickly take a minute to introduce everybody to the PETA Science Consortium International. For all of those who are not familiar with this organization, the Science Consortium was established in 2012, and it comprises of more than 20 scientists from all over the world, from India, from US, UK, Germany, Netherlands, Australia. And the Science Consortium basically works to promote the regulatory acceptance and implementation of non-animal methods. We do this by funding the development and validation of test methods. We also have a very comprehensive list of the different alternative methods which are validated for regulatory purposes at our website. We also organize free training opportunities and host workshops and webinars for interested regulators, companies, and scientists. I will highly recommend all of you to please go and check out our website and do see the training videos and webinars that are available in case you're interested in understanding more about how these non-animal methods are being used for risk assessment purposes. Apart from this, we also do a lot of retrospective analysis of the existing test method to demonstrate their utility and their usefulness in risk assessments. Apart from that, we do a lot of outreach. We present and publish our work, attend scientific conferences, and connect with like-minded people to facilitate replacement of animal tests wherever possible. On this note, I would just end my presentation here. There are just two excellent opportunities that I would like to highlight here. The first thing is that PETA India is looking to hire for the position of a science research associate. And interested people who have relevant experience in toxicology, biochemistry, molecular and biotechniques. And if you're interested in using your knowledge and experience to help animals, to help replace and reduce animal testing, please do apply for this post. And apart from that, I am happy to announce that PETA India is inviting applications from early career researchers who are working in the field of alternatives for a travel award. It's an international travel award. And just please note that the last date for submitting applications is 31st December. And anybody who has any questions about any of these opportunities, please feel free to get in touch with me at the mentioned ID. On that note, I would like to thank everybody once again, and I'm happy to answer any questions. Thank you so much.
Thank you, Dr. Ankita Pandey, for your elaborative lecture on the activities of PETA India as well as the pesticide safety assessment. You very well defined many non-animal approaches, top-down approach and bottom-up approach and integrated approach for toxicity of the laboratory of for the testing of pesticide. So I think we can take one or two questions from the audience. And if there is any question for Dr. Ankita, then she'll be happy to answer. Okay. If there is no question, can I ask one thing, like if these toxicity testing for pesticides would help in generating environmental risk assessment? These tests would help in environmental risk assessment in generating an ERA?
Absolutely. I mean, the weight of evidence approach that I've mentioned applies equally to human health risk assessments and ecotox assessments. And if you talk about in vitro models, we do have different cell lines. We have, for example, yesterday somebody spoke about the cell line that's been recently validated for fish toxicity testing. So we do have a lot of work being done in developing alternatives for ecotox assessment. But the standard one would be using a weight of evidence approach before directly testing your test substance on these animals.
Thank you, Dr. Ankita.
Can I ask you one small question?
Yeah, Dr.
I mean, it's just a comment, not a question. Basically, that the EPA. Somebody is talking behind.
Can I request Dr. Rohit Seth with mic is on? Okay, thank you.
So if you follow the environmental toxicology for the last 40, 50 years, there's a lot of evolution happened from starting with animal tests by EPA and other organizations. Even the universities with the Toxic Superfund have got involved trying to characterize what are the minimum tests that you can do. Today, AOP pathways are overkill, as a lot of people are doing it. They're doing toxicogenomics, this, that, everything they're doing is trying to get a lot of data into it. But recently, there's this big change that I've been seeing. In fact, one of the professors I worked with at the University of California, Berkeley, he and the Environmental Toxicology Consortium that they have with NIEHS, they're promoting something called key characteristics. Key characteristics are only 7 or 8 characteristics, whether it is an endocrine disruptor or a carcinogen or a skin carcinogen or something like that. They have only 7 or 8 key characteristics they're identifying with all the available data that's been there for 50 years now. Right? I mean, but the EPA and all these people are still reluctant to use that information because we are doing tons of work. Nobody knows how much data we are generating and what is the relevance to the human. At the end of the day, all this should be relevant to the humans. And that I'm not seeing it, that we are all doing all kinds of things, but not showing the relevance to the human. Any comment from you, Ankita?
So as far as my knowledge goes with respect to EPA, I would say that they have been quite receptive and open towards the idea of reducing animal testing. If you would know, the last administrator, Andrew Wheeler, he even came up with this memo saying that by 2025, they would cut down mammalian testing by 25% by 2035. They would end mammalian testing, the funding and request, and they have like a work plan in place to ensure that that they implement this memo. We have like the PETA US, our entity at US, they've been working very, very closely with US EPA on this. And for what our experience has been, they have been quite receptive towards this issue. For example, we are working with them to conduct a lot of retrospective analysis. And based on those analyses, they have been accepting a lot of waivers, like giving you a perfect example, if you talk about ecotox assessment, just like India, even US EPA requires 2 tests conducted on birds, the acute study and the repeated dose study. So PETA US closely worked with US EPA. We looked at all the agrochemical ingredients that were registered from 1998 to 2018. We did an analysis to see whether both the tests are required for driving regulatory decisions. And what we found was that we compared the risk quotient values from both the tests. And what we found was that the acute oral study was enough to drive regulatory decisions. And there was not even a single chemical based on which the acute oral study was not giving your higher risk quotient value. And on the basis of that study, US EPA finally set up a waiver saying that if you have a study available from acute oral for the bird study, and the particular test substance is not expected to be bioaccumulative in nature, in that case, the repeated study may be waived off. So as my experience, of course, you've been in the industry, you've been working for many, many years than me, and I am comparatively more new to this, but we think that US EPA is gradually changing, and they're becoming really receptive towards this cause of.
I think there is the academic excellence is there, that they have to recognize it, and academic excellence really is super. And EPA is any regulatory agency is very slow to make the modifications to the regulations, and hopefully this will change. Okay, all right.
Okay, so thank you once again, Dr. Ankita Pandey, for your wonderful lecture. So now we will move to another distinguished speaker, and we'll try to hear from Dr. Rakesh Kumar Singh from NIPER Bareilly. And for our audience, I would be happy to introduce Dr. Rakesh Singh. Dr. Rakesh is basically working as an associate professor and head of the Department of the Pharmacology and Toxicology at NIPER Bareilly. His broad research area includes translational research on the molecular pathways involved in inflammation, in neurodegeneration disorders, airway disorders, and chronic autoimmune diseases. Dr. Rakesh, basically, he did his PhD from the Jamia Hamdard, New Delhi. So must be a student of Dr. Raisuddin, and MS or the in pharmacy, in pharmacology and toxicology from very prestigious institute, NIPER Mohali. So he has over 11 years of now the rich industrial research experience in vitro pharmacology and new drug discovery. During his tenure as industrial scientist in new drug discovery, he worked on exploration, selection, and validation of novel therapeutic targets in the broad area of chronic airway disorders and chronic autoimmune inflammatory diseases. In addition, he had been instrumental in planning, budgeting, and implementing new scientific programs in collaboration with other departments. Seems very active and very cooperative to the scientists, to the other scientists. So I now welcome Dr. Rakesh Kumar Singh for your wonderful lecture on the importance of cell-based models in phenotypic drug discovery. So Dr. Rakesh.
Thank you. I hope I am audible to all of you.
Yes, you are, and your presentation is also visible.
Okay, thank you, Dr. Aman, and thanks a lot for the nice introduction which you have portrayed. Just to correct you, I am a PhD. I was a PhD student of Professor Najmi from Department of Pharmacology at SPER, School of Pharmaceutical Education and Research. Okay. But of course, Professor Raisuddin has helped a lot in my educational career. So you caught it right. One way or the other, I know him well, and he also knows me well. So I thank you. I thank the organizers from DUPSU, Professor Patil, the members, executive members from the society, Professor Raisuddin, Professor Akbar Shah, for giving me an opportunity to talk in this eminent conference, in between the eminent personalities. So moving ahead with my presentation, the topic of my presentation is importance of cell-based assay in phenotypic drug discovery. Now, we have been talking about cell-based assays or the models alternative to animals since yesterday. And I think I would not talk much about that because we have been talking much on this topic since yesterday. So we will look at a bit of cell-based assay, how they help in discovery programs, particularly in phenotypic drug discovery. And we will also try to understand what is phenotype, why we say this as a phenotypic drug discovery. Yes, so the cell-based assays, as I said, they have a very much potential relevance in early drug discovery process. And there are many types of cells that we use, like human primary cells, cell lines, or the monocultures, co-cultures, or the mixed cultures like neurospheres, which are composed of a mixture of progenitors, microglia, neuronal cells, etc. There has also been a few talks on 3D, 2D bioprinting, 3D bioprinting. I heard those talks yesterday. And these models have provided relevant information with respect to the advanced technology which have developed in recent years. Like they provide multi-parametric high content imaging techniques are there through which we can have like high content data. There are more data collected together at a time using those images from cells. And the assays can be designed to study the complete signaling network inside the specific cells. Now, these signalings can be either cell-cell interaction. There can be either protein-protein interaction, the biomarker studies that can specifically be done by designing suitable cell-based assays. So cell-based assays are a good alternative to animal models that provide relevant information in drug discovery. Now, what are different types of assays that we do by using these cells? There are certain routine studies that we do, like the common assays which we do are cell proliferation, cell viability, cell morphology, and motility. We can also look at the downstream signaling pathways, which happens through activation of different enzymes or kinases as therapeutic targets, like, for example, the phosphorylation or dephosphorylation events, and can also look at the deciphering the mechanism of action by looking at the downstream mediators. And apart from kinase and enzymes, we can also look at the receptor-based studies by certain binding assays. One of the good example, classical example, is radioligand binding assays that still we do. And the functional assays, like where we look at signaling mechanism, the downstream signaling mechanism with respect to G protein coupled receptors, like the functional assays which we do, cyclic AMP, calcium, and they provide relevant information about affinity as well as intrinsic activity of drug to the receptor. So how should all these cell-based assays be? Like an ideal cell-based assays, when we say they should be very simple to perform, they should be non-radioactive, they should not involve any radioactive materials. Like, as I said, we use mostly radioligand binding assay for GPCR study, and still we use today. But an ideal cell-based assay should be non-radioactive to avoid any chances of radioactive exposure. It should be robust and reproducible. It should be homogeneous. And most importantly, it should be amenable to microtiter plate format for robotic automation to increase the throughput. And it should be cost effective. So these are certain characteristics that should be there in an ideal cell-based assays. Looking at this, we should try to follow as close to these parameters as possible. Now, moving on to the different types of drug discovery approaches using cells. Like, as I said, there is a phenotypic drug discovery, which has come up in recent years. And classically, there is a target-based drug discovery that we perform. Now, there is a difference between the approach which we follow in both these types of drug discovery. Like when we say target-based drug discovery, the target-based drug discovery identifies the compounds that produces a biological response on an isolated target. And we know about the target in very early in the stage, in the preclinical stage. Whereas in case of phenotypic drug discovery approach, we do not know the target firsthand. And this is not classical approach that we follow. Here, first we look at a compound which can produce a biological response in a cell-based or either in a cell-based model or in an animal model. And then once we see the desired biological response, then we try to identify the target and validate. So the target identification is at a later stage as compared to the target-based drug discovery. However, both these are the parallel pathways to achieve the same goal. Now, we know that drugs typically act by engaging a molecular target. However, in phenotypic drug discovery, a prior knowledge of that target is not essential. We emphasize more on the physiologically relevant biological system or the cellular signaling pathway. But the important thing here is we must know in a particular disease which physiological relevant pathway or cellular signaling plays an important role, so that we can look at a relevant biological endpoint. And that can be directly interrogated to identify biologically active compounds. And once we identify a biologically active compound, then we'll go for target deconvolution approach. We decipher that, we go ahead to work on the target. So there are certain strengths and certain disadvantages of target-based drug discovery. Like, as I said here, the target, in case of target-based drug discovery, the target identification is at a much earlier stage. Once we finalize a disease to work on, and then we go ahead with target identification, and the rest of the things is in line with the target signaling, with that particular target. And then we go ahead for preclinical discovery and development, and like that we go. So the strength of this is we can apply the molecular knowledge which is already existing in the literature to investigate the specific hypothesis for either a small molecule screening or the biologic-based molecules. But the disadvantage is like a specific molecular hypothesis which we formulate during disease selection and target identification may not be much relevant to the disease which we select, to the disease pathogenesis, or may not provide a sufficient therapeutic index. So there are certain other limitations also. Like, for example, traditionally we know that several industries, they follow this target-based drug discovery approach. And now we know that the graph of discovering a new drug is going down. There has hardly been any new drug that has been discovered in recent years, small molecules. There has been increasing failure at the phase 2 or phase 3 due to lack of predictivity of translational in vivo efficacy in early preclinical stage. And there is highest drug attrition rate primarily in the clinical stage, which is the most expensive stage for a drug to fail. So looking at all this, we know that there is a decreasing productivity, which has prompted a resurgence in the phenotypic drug discovery approach. Meaning we need to first look at the biologically relevant endpoint. We need to look at whether our drug molecules, they have any effect on those biologically relevant endpoint. And then we should go ahead to identify the target and target validation. This is one of the recent data, and this data shows that the big multinationals which are there, like for example, in terms of expenditure, R&D expenditure, if we see, the Merck, BMS and Ross, which are the top three companies who has invested much on their R&D, like as we can see here, $13 billion, $12 billion, and $11 billion. And these companies has, if you look at the new drug which has been discovered by these top 20 companies, there has been an inverse correlation to the R&D productivity. Even in the era of what we say is advanced screening technology. Today we have much advanced screening technology, but with that reciprocation, the R&D productivity has not increased. And one of the reason is probably the target identification is at first place, which may not be connecting to the relevant biological endpoint correctly. So that could be one of the reasons. So that's why we say that the phenotypic drug discovery is one of the more relevant approach that the company should follow, or the people involved in the drug discovery should follow, in order to have a successful innovative drug. So the strengths are we do not require a prior knowledge of mechanism of action, and the target identification is not here at the first place. If we look at the steps involved in phenotypic drug discovery, the target identification and target deconvolution comes at a very later stage, after we get a hit compound or a lead compound, then we can go ahead with the target identification. And the activity in such assay might be translated into therapeutic impact more effectively than in the target-based drug discovery. However, there are certain disadvantages also, like the challenge of optimizing the molecular properties of drug without prior knowledge of mechanism of action may be a challenge. There is a considerably lower throughput than target-based drug discovery approach. And that we all know, although there is no difference between the expenditure involved as well as the time involved in between the target-based drug discovery and the phenotypic drug discovery approach. Another example which I found is between the target-based drug discovery and phenotypic drug discovery is target-based drug discovery. It's best to find a best-in-class drug. It is not best to find a first-in-class drug. So we can follow this approach to find a follower drug or for a best-in-class drug, but it is not suited for first-in-class drug. For first-in-class drugs discovery, we should follow phenotypic drug discovery approach. If you look at the data in last 10 to 15 years, the first-in-class drugs which were discovered were more by phenotypic drug discovery approach as compared to the target-based drug discovery, which is completely opposite for a follower drug or for a best-in-class drug. So there is a difference which we see practically in terms of discovery of first-in-class as well as best-in-class drug in terms of these two drug discovery approaches. Now, moving ahead, there are certain other advantages also why we should follow the phenotypic drug discovery approach. In target-based drug discovery, we stick to a particular target and we test our compounds with emphasizing this particular target. But in phenotypic drug discovery approach, we have a library of different scaffolds, and that can be tested on different targets, maybe one by one, or if we have a system that can express all these targets, we can screen subsequently and measuring different endpoints, different biological relevant endpoints with respect to which are connected to this particular targets respectively. So we can look at different pathways together, and then we can go back to target validation, basically, that probably one compound is signaling through this particular target and giving this particular biological endpoint. So there is this particular approach imparts a much larger molecular target space to work on. Now, there is another term which we say is polypharmacology. So that's what we discussed here. Like if we look at this particular approach, we can look at subsequent targets signaling pathway, like different pathway can be studied subsequently at a time using this approach. And this approach we say as a polypharmacology approach. Once we have identified a particular signaling, and then we can go back to target deconvolution, and then individual target can be studied and validated by this approach. Now, this approach can also provide a mechanistic bridge between the disease pathophysiology.
Dr. Rakesh, could I request you to please try to conclude your lecture, please, Dr. Rakesh?
Yes, sir. Yes, sir. So, like if we have different compounds which works on different targets, then we can identify the molecular phenotype depending upon the signature shown by different compounds. And then we can go ahead with the in vivo validation. Moving ahead, there are safety aspects of these projects also, like there are certain off-target safety risks and there are on-target safety risks. So the off-target safety risks are like selectivity data, again, against closely related proteins we need to look at. There should be like a structure related, there can be a structural related issues which can be predicted with the polypharmacological approach, and they are non-specific. There can be non-specific or non-selective issues. There are certain on-target safety risks, like the target expression pattern in cells and tissues may be different. The phenotype of target deficient or knockout models should be known prior to this, and the phenotype of human target specific mutation. Like for example, if we identify a target, we need to know its particular phenotype. So this approach has contributed to development of commercially successful first-in-class therapeutics for unmet medical needs. During the last 10 years, the PDD uses has grown from an estimated 10 to 40% of project portfolio of the selected companies. And it has been shown in literature that the big multinationals, they have published the PDD, and this contributes approximately to 25 to 40% of their respective discovery profiles. So if there are any questions related to this presentation, I would be happy to answer. Probably I will not take much time to finish this.
No, it's okay, Dr. Rakesh. I think it would be better if we can ask we can take one or two questions.
Yes, sir.
That's good. If there is any question from Dr. Rakesh, please. The forum is open for any question. So if there is no question, I would like to thank Dr. Rakesh for this wonderful lecture, and he tried to present the ideal-based, cell-based assay in the phenotypic drug discovery, which has potential in the drug discovery. And he very well explained the difference between the phenotypic drug discovery and target-based drug discovery, and the advantages and disadvantages of both these. And he tried to explain that phenotypic drug discovery is more relevant in this era. So thank you very much, Dr. Rakesh, for a wonderful lecture.
Thank you.
So
thank
over
you.
to Dr. C.R. Patil.
Yeah, thanks a lot, sir. Thanks for sharing this session, and I'm thankful to both the invited speakers, Dr. Ankita Pandey and Dr. Rakesh. It was really nice to listen to both the invited speakers. We are thankful for your time, sir. Thanks a lot for joining us for this presentation.